I just want to welcome everybody for joining us. And we're so excited today to have the Tacer Lab here presenting. And I would like to introduce all of you, our USP7 family, to the lab. And I don't know who's presenting first. Klementina? Okay, so we'll let Klementina go ahead and start.
Awesome. Can I share the screen? Yes, I can. Okay, so do you see my screen now well?
Yes, perfect.
Wonderful. I will remove that. So thank you for inviting us. So we are very excited to present what we do here and how we structured today. So today we are 3 members of my lab are here. Tara Bayat and Denish Tepihar joined me. They are graduate students in my lab. What I will do, I will just briefly introduce our lab and work that we do, and then I will let them speak about their projects. And we try to make it in a way to present in a more lay language a little bit, but you are welcome to ask if we are not clear. So as said already, I'm Klementina von Tacer. I have my lab in Amarillo, Texas, at Texas Tech University School of Veterinary Medicine, where we also have a Texas Center for Comparative Cancer Research. So we try to combine the different aspects of our research to understand what is going on in patients and also in animals. And let me see, okay, we need to do that. So maybe just to get us started, where are we located? We are in Amarillo, Texas. So this is where this big star is. And for those of you who may not know Amarillo, Amarillo is just in the middle of Route 66. So in Texas Panhandle. And this is how our school looks like. It's a new School of Veterinary Medicine school, and it is part of Texas Tech University that is located in Lubbock, the main campus, but we are here in Amarillo, Texas. And what we do in my lab is we are trying to understand how we react to stress and also how we protect against stress and how we do that on the whole body level and on cellular level. And we are in particularly focusing on how germ cells in the testes are protected. How is this hijacked by cancer? And I will let you know a little bit why is this relevant. And the most important for today's talk, we are studying also how some processes are disrupted and may be relevant to the neurodevelopmental disorders like Hao-Fountain Syndrome. And what connects all these projects that seem very diverse is the proteins that we are studying and are called melanoma antigen genes. And what our data started to show and unveil is that it seems like that these proteins evolved to help protect our cells against different stressors and help them adapt to changes in environment, because this is what we need to do. Environment changes all the time. Food availability changes, temperature changes, different people contact changes, and we need to adapt. And there are several mechanisms that evolve that help us do that. And we are finding that these proteins manage these processes to be even more faster and more useful for our bodies. And how are these proteins discovered, these melanoma antigens or MAGEs in short? they were discovered as tumor antigens. These proteins are the first tumor antigens ever discovered on tumors. MHA1 is now called this. And what are tumor antigens? These are these proteins on the top of cancer cells that signal, I am not yours. And our immune system in our body recognizes, like depicted here as this, one of the immune cells, T cells, recognize these proteins and knows, oh, I have to attack that, this is not mine. And this really attracted a lot of attention in 1990s. And it's still now one of the major mages are one of the major targets in cancer immunotherapy. And interestingly, what we found is that these proteins that are very commonly expressed in cancer are normally in healthy males expressed in the testes. That's why we are interested what they normally do. And these proteins are very interesting from evolutionary point of view. They are present in all eukaryotes, we say, in all organisms that have cells with nucleus. And however, most of the animals have only one gene. But then with monotremes and in particularly with placental mammals, these genes expanded to 40 or more genes that we have in our genome, but also other mammals like Texas longhorns, cattle is one of the animals that have a large number of these genes. And we are interested what they do. What I'm showing you here is phylogenetic tree, as we call it, of human mages. And you can see they come in two different flavors. We call them type 1. These are the red and blue ones. These are the ones relevant in cancer. But today, for today's talk, we are really interested in these green ones, and in particular in this MHL2, because this is the protein that works together with USP7. And Tara and Dennis will tell you more about this, what we know so far and what we want to discover. And these proteins are often in the brain. And that's why we are also interested in MAGE function in the brain. And this is just a summary of a lot of my work depicting exactly what I told you so far. These MAGE genes that are in red or here in yellow and even in green are highly expressed in cancer and in germ cells, in testes, even in ovary. But in the brain are these type 2 mages, we call them, and MHL2 is here. And we are trying to understand what is MHL2 function, in particularly in hypothalamus, and how is this related to Prader-Willi syndrome and Schaff-Yang syndrome, because this is the gene that was discovered to be involved in these two neurodevelopmental disorders. And you know that these children with Prader-Willi or Schaff-Yang, they present several similar symptoms like Hao-Fountain syndrome. And actually, by investigating Prader-Willi and Schaff-Yang and the role of MHL2 in these two syndromes, we also discovered that Hao-Fountain syndrome is related to the similar processes. So now I will give the podium to Tara and then to Dennis to tell us more what they do and how they are trying to understand the role of MHL2. So with this, I will stop sharing my slides and ask Tara to start. But I can also
Thank you, Dr.
address any questions if they are earlier.
Do you have my screen?
Yes.
Yes, okay. Good.
Just, yes, share it as a screen.
Well, thank you so much, Dr. Tacer, and thank you everyone for listening to our talk today. My name is Tara Bayat. I'm a PhD candidate and graduate research assistant in Dr. Klementina Tacer's lab. I'm very happy to talk about our project today along with Denis. So first, I would like to give you an intro about USP7 and why is it important in our project and how it is connected to our project. Ubiquitin in a specific protease 7 or USP7 that was originally known as herpes virus associated ubiquitin in a specific protease 7 or HOSP is a protein coding gene which is located on human chromosome 16. Let me go to this. So it is helpful to mention the central dogma of molecular biology here because we're going to go back to it later when I explain my project. So as you know, in our genome is DNA, and how DNA is translated to protein has a process that is explained in central dogma of molecular biology. First, DNA is transcribed into RNA, and we have different types of RNA. And one of them is mRNA, which goes through mRNA translation, and through mRNA translation, proteins are synthesized. And of course, USP7 is a protein, which is coded by USP7 gene. And on an organismal level, USP7 is involved in several homeostatic mechanisms in the body. For example, DNA damage response, immune response, tumor suppression, and epigenetic control. However, it's involved in different mechanisms on a cellular level as well. So if you want to look at the USP7 functions on a cellular level, we should mention one of the most important ones, which is the ubiquitination. So what is the ubiquitination? Ubiquitin is a very small protein in the body which binds to proteins through the function of some enzymes called E3 ubiquitin ligases. So E3 ubiquitin ligases are enzymes that bring ubiquitin molecule to different proteins, like many proteins in our body. And what does this ubiquitination do? It might cause the degradation of the proteins, or it might activate different functions in the body, which are crucial in our signaling pathways. So on the other hand, there are enzymes which are called the ubiquitinating enzymes, or DOBs. And USP7 is one of those, the ubiquitinating enzymes, which remove the ubiquitin molecule from the proteins, and it plays crucial roles in different mechanisms in our body. So one of these mechanisms is through a complex called MUST complex, where USP7 binds with MAGEL2 and TRIM27. And so it creates, together, three of them creates a complex called MUST complex. So TRIM27 is an E3 ubiquitin ligase that I mentioned in the previous slides that bring the ubiquitin molecules to the other proteins. And MAGEL2, that we're going to talk about it more during the presentation, is very important for the function of TRIM27 and USP7 in this complex that fine-tunes the activity of TRIM27 for ubiquitination. And so together, MUST complex regulates the ubiquitination of a protein called WASH. And what is WASH? So WASH is important for regulation of hormone secretion. So this MOST complex, which is composed of MAGEL2, USP7, TRIM27, as I mentioned, is important for regulation of hormone secretion in our body, as they regulate protein trafficking and protein recycling in the body. And so we know that hormone secretion is crucial for many homeostatic mechanisms in our body, and is important for our responses to many environmental stressors. But this hormone secretion is particularly important in hypothalamus. We know that hypothalamus is a region in the brain which is critical for several physiological and homeostatic functions. For example, sleep, metabolism, food intake, body temperature, growth, and also response to stress. So in Hao-Fountain individuals, as well as two other disorders called Prader-Willi and Hao-Fountain syndromes, many of these homeostatic mechanisms are dysregulated. And what are Prader-Willi and Hao-Fountain syndromes? These syndromes are neurodevelopmental syndromes similar to Hao-Fountain, which are caused by a deletion or mutations in MAGEL2 gene, which was one of the binding partners of USP7 in the MOST complex. So studies have shown that several hormones and neuropeptides are dysregulated in these individuals. And those hormones are essential for these physiological and homeostatic mechanisms, particularly in the brain. And this dysregulation causes the phenotypes of the disease, such as autism spectrum, growth retardation, infertility. And these are also seen in the MAGEL2 knockout mice that we are studying. So as I mentioned, Prader-Willi syndrome, Hao-Fountain syndrome, and Hao-Fountain syndrome share several phenotypes, and many of these phenotypes suggest hypothalamic dysfunction. And these phenotypes include autism spectrum, developmental delay, intellectual disability, and hormonal imbalance. So in my research, we aim to determine the role of MAGEL2 in regulation of mRNA translation. So just to quickly remind you of the central dogma of molecular biology that I mentioned quickly in the previous slides. So mRNA translation is the important step where mRNA is translated into polypeptides and protein. This is how all the proteins in our body are synthesized. So this is a very important step in the regulation of gene expression. Now we want to see if MAGEL2 is involved in the regulation of this mRNA translation in the brain, particularly in the hypothalamus. So to address this question, we performed polysome profiling in the hypothalamus and pituitary gland of MAGEL2 wild type and knockout mice. So we are studying, our model for studying this is our mice. We have normal mice, and we have the mice which are MAGEL2 knockout. And we collect hypothalamus and pituitary gland, which is also another gland that is important for hormone secretion and response to stress. And we perform polysome profiling on these tissues, which is a complicated technique, but just to give you a clue about the aim of this technique is to see how individual mRNAs are translated and what's the speed of their translated, because it's important to see if MAGEL2 deletion causes suppression or activation of the other genes in the hypothalamus and pituitary gland. And by other genes, I mean many of those hormones and neuropeptides which are dysregulated in patients. So this is a polysome profile of MAGEL2 wild type and knockout mice from pituitary gland that we got, and it shows different steps of mRNA translation and their associations with ribosomes. So we perform fraction analysis, RNA isolation, and then RNA sequencing to see which genes are differentially expressed and which genes are dysregulated in the hypothalamus and pituitary gland upon loss of MAGEL2 in these tissues. So from our analysis, as you see the numbers, we got a lot of genes, a lot of transcripts which are dysregulated upon loss of MAGEL2 in the hypothalamus and pituitary. And it suggests that many of those transcripts are regulated at the translational level. And it suggests the role of MAGEL2 in regulation of mRNA translation and protein synthesis in the hypothalamus and pituitary gland of Prader-Willi and Schaaf-Yang individuals. So this is important because the condition in our experiments, they were a normal condition. They were not exposed to any type of stressors like heat shock or like fasting. These mice were under normal condition, but they were MAGEL2 knockout. So it suggests that even MAGEL2 knockout mice are stressed under normal condition. And it suggests the role of MAGEL2 in regulation of stress response in the brain, which is very important to address it in not only Prader-Willi and Schaaf-Yang, but also in Hao-Fountain syndrome to see if MAGEL2 is involved in the regulation of a stress response through its connection to USP7. So recently, we also developed iPSC-derived organoids from the patients. However, and I'm showing this picture, which is a cerebellar organoids on the day 22 of maturation. And we are growing them. However, these are not from the Prader-Willi or Schaaf-Yang patients, but these are other models. But we are developing these 3D models from the patients in our lab. And this is very interesting to also study them and develop these 3D models from Hao-Fountain patients in the future and collaborate. And so this is also a very useful in vitro model, as our work focused on in vivo model so far, but we are moving forward with these in vitro models as well. So thank you so much. And now I will pass it to Dennis to talk about his part of the project. And I can stop sharing if you want. So Dennis will share.
Yeah, perfect. Okay, do you see? Is the screen okay? Everything okay? Perfect. So yes, my name is Denis Tepihar. I am also a graduate student in Dr. von Tazer's lab. I would like to thank Dr. von Tazer and Tara for their part of the presentation. And now I'll focus on my part of the project. If you remember from the central dogma of biology, molecular biology, Tara was focusing on translation, whereas I'm focusing on transcription, so the process from the DNA to the RNA. And our project had the aim of determining the role of MAGEL2 in the hypothalamus on a single cell level. I'll explain what that is in a bit, in normal fed and fasted states. So we exposed these mice to some sort of stress, which was in this case fasting. Here, just to briefly remind you the role of MAGEL2 and its interplay with USP7. So MAGEL2 binds to the endosome. These endosomes, they're cellular compartments that are important for protein recycling and protein trafficking. And to MAGEL2 binds TRIM27, which ubiquitinates WASH, which then promotes this protein trafficking. And TRIM27, ubiquitinates, and USP7 binds to MAGEL2 and does the exact opposite because it's a deubiquitinase, so it deubiquitinases WASH and also deubiquitinates TRIM27, so it doesn't degrade itself, and in that case, fine-tunes this whole mechanism of protein trafficking and protein recycling. So this single cell sequencing that I promised I'll explain, MAGEL2 is expressed most highly in the hypothalamus, and the hypothalamus is very important for the regulation of homeostatic mechanisms, as Tara mentioned. Think temperature control, fluid control, hormone release. And this tissue consists of several different cell types. Some studies report from 30 to 60 different cell types. And these different cell types have different functions and contribute to different processes. And we want to understand how these specific singular cellular populations change when we, for example, knock out MAGEL2 or expose these mice to stress. And for that, we opted for the single cell approach. And the single cell approach is the most easily visualized if you imagine you're making a smoothie. So this fruit bowl, imagine, is your hypothalamus. You have different fruits in the fruit bowl, which represent different cell types. So if you opt for the classic bulk RNA sequencing approach, imagine you just blend all of these cell types together, and what you'll get is the average gene expression across all cell types in this specific tissue. But what we opted for is the single cell RNA sequencing approach. In our case, it was single nucleus. And what we get here is, imagine testing all of these different fruits individually in this fruit bowl. And with that, we can identify rare population types. We can define different cell populations and how these populations change, and also changes within these populations, which is, of course, very important in an organ that is so heterogeneous as the hypothalamus. So for the actual practical experimental approach, we used, again, C57 black 6 mice, both wild type, so normal mice, and MAGEL2 knockout mice. We used 4 mice per group in 4 groups. So these groups are MAGEL2 knockout. These knockout groups are just mice that we knocked out MAGEL2. And wild type groups, so these are normal mice. And both of these groups were also split into fasting and control groups. So fasting groups were fasted 24 hours before the experiment, and the control group was just given food as normal. After this 24-hour fasting, we collected the tissue, performed tissue dissociation, captured these single nuclei, these single cells, sequenced them, and then came the data analysis. We performed quality control, alignment, quantification, normalization, dimension reduction, clustering, cell type annotation, and then finally differential gene expression. And these differentially expressed genes, I'll explain what that is in a bit, we used to perform gene ontology, pathway enrichment, and gene set enrichment. So here's what we have found. The hypothalamus, as I mentioned, very heterogeneous. And through this different expression profiles of these different cell types, we identified 41 distinct clusters in our data. And then we used the different cell type markers. So think each cell type has one or usually more markers that mark it as that specific cell type. For example, if we look at oligodendrocytes, they express PRR5L, and through these markers, we can actually assign a name to these different clusters and define different cell types. And through using these markers, we identified 23 distinct cell types. For our ease of understanding, we marked all neuronal cells in green, in these 2 shades of green, and non-neuronal cells are multicolored. What is very interesting, first of all, is that more than 66% of all cells that we identified were neuronal cells. And this is somewhat what you expect when you're working with hypothalamic data. But when it gets very interesting is when we look at this graph here. I know it's kind of a nightmare, but we'll go through it. So here on the left side, we have the number of differentially expressed genes. So these differentially expressed genes, these just represent the genes that change when you compare two groups. So for example, here in red is knockout control, so the hypothalamus of a major to knockout mouse, and wild type control, so a normal hypothalamus. And when we compare these two groups, we get a specific set of genes that are changed. And we associate these genes with what happens when we knock out MAGEL2. And what we see here is that if you remember, more than 66% of cell types were neuronal, but most of the differentially expressed genes were actually in non-neuronal cell types. For example, here, endothelial cells, microglia, tanycytes. Then we built Venn diagrams using these different comparisons and the differentially expressed genes. Here, I'd like to call your attention to this group here in red, this 64. This is the comparison of a knockout control hypothalamus and a wild type control hypothalamus. So this is what happens when we knock out MAGEL2. And this group here in green, this 26 here, and this is the comparison of a wild type fasted hypothalamus and a wild type control hypothalamus. So this is what these genes are, the genes that change when we fast a normal mouse. And what you'll notice here is the overlap between these two groups. The overlap between a MAGEL2 knockout hypothalamus and a wild type fasted hypothalamus is quite significant in several different, in most different cell populations. And what this leads us to believe is that MAGEL2 is critical for adaptation to fasting. And and the thing that Tara was mentioning before is these mice, these MAGEL2 knockout mice, even when not exposed to any stress, they seem like their hypothalamus thinks that they're under stress. It thinks that they're hungry constantly. And to illustrate this point a bit more, we also performed gene ontology. So gene ontology is just a very large database where they assign different terms to different genes. For example, protein. For example, you have one gene and you assign protein localization to synapse, chemical, postsynaptic transmission. And what we compared here in blue is fasting in knockout animals, and in green fasting in wild type animals. And what we see here is that these terms, they don't overlap much. And what that leads us to believe is that the actual response to fasting in knockout animals versus wild type animals is significantly different. And interestingly, we also identified a set of uniquely enriched neurons in both the fasted hypothalamus, so the wild type fasted, and MAGEL2 knockout hypothalamus. And these are IQ-gap2 expressing neurons. If you look here, you have wild type fasted hypothalamus. These are cell type fractions. So in wild type control, these IQ-gap neurons are present in a very low amount. But when we fast these wild type animals, the percentage or fraction in this case of these IQ-gap2 expressing neurons rises. And we see a similar phenomenon when we have MAGEL2 knockout animals. So these two are quite similar between each other. But what is the most interesting in this is when we fast MAGEL2 knockout animals, and in this case, this percentage of IQ or fraction of IQ-gap2 expressing neurons skyrockets. And why is that so interesting? If we look from the perspective of the MAGEL2 knockout, this IQ-gap2 protein, if you remember this image that I showed you before, this IQ-gap2 protein interacts with this WASH protein and promotes F-actin nucleation, which then promotes or regulates cell protein trafficking and protein regulation. And if you look from the fasting perspective, it can be a signal molecule and affects the insulin and glucagon pathways. So just to briefly conclude, the hypothalamic response on the single cell transcriptional level of C57BL6 MAGEL2 knockout male mice mimics the wild-type fasted state, and IQ-gap2 expressing cell types are enriched in both knockout control and wild-type fasted hypothalami, which suggests its role in the adaptation to nutritional stress and possibly the compensation for the loss of MAGEL2. Also, MAGEL2 seems to be critical for a normal response to fasting, and its depletion renders the hypothalamus towards a more fasted, so more stressed state. I would like to acknowledge first all of you from the USP7 Foundation for giving us the chance to present our research, and then our PI, our advisor, Dr. von Tacer, and all of our lab members, and also all of our collaborators from Dr. Ponomarov's lab, Dr. Karamisho's lab, and Dr. Karamisheva's lab. And with that, I will conclude my presentation, and we are open to any questions you might have.
Thank you so much. That was so interesting. I think for me, one of my questions is because I'm not a scientist, you guys will know better than me. So because of MAGEL2, like those disruptions, and our kids have the USP7 disruption, so would they like have similar kind of experiences with those like in their hypothalamus, you think, because of the USP7? Is kind of what I'm gathering, like it would be a similar type thing with like the stress and whatnot.
If I may to start, that's exactly what we hypothesize. And also we are very interested to check whether or not MAGEL2 is changing in the neurons of USP7 patients. And that's why these models that Tara was showing are so critical. So what we can do then, get fibroblasts from your patients, from your children, make them in the dish like neurons, and see whether or not they have what is the level of USP7 and how this contributes to MAGEL2, because what we know from molecular level, these three proteins need to be together. If they are not together, MAGEL2 is not stable. But I think that's a missing part that definitely we would like to address, to see how much MAGEL2, because at the end of the day, what we would like to know, how much of the phenotype depends on MAGEL2, and that is very important because the treatment that we can find for Prader-Willi, Schaaf-Young will also benefit your children. On the other hand, of course, it's very interesting also what is what are MAGEL2 independent functions that are critical for your children that we are missing in your children. You know, USP7, and we didn't show that, but we know that USP7 is expressed in every cell, and also it's expressed at quite high level, and it's a protein with a lot of function, as Tara mentioned. MAGEL2 is expressed much more selectively. The highest level of MAGEL2 is in the hypothalamus, also in placenta. What we are also very interesting, very little is known, like during embryonic development also. Could we do, like if we diagnose this earlier, can we help earlier, even before these children are born? These are kind of projects that we have in mind for the future. We don't have it yet, but are very important, I think.
Yeah, it's super interesting. And just to hear like about the stress level, and I don't know how it kind of correlates, but I do, like with my daughter, with Ella, she is very stressed a lot, like in general, like very high anxiety. Things stress her out that normal people would not be stressed about.
Exactly, and that for us was so interesting, both of like type of assays that we do, because when I started my lab and when Tara and Dennis joined, we introduced the stress component to our research exactly for that purpose, because of our other projects for these MAGE proteins in the testes, for example. What we found, like we have 10 MAGE-A genes in germ cells, male germ cells. We knocked out all of them, like deleted all of them out. and males are okay, and they still are fertile. But then we start to think, why would you have so many genes for no reason? And then we started to think, oh, we keep our mice in five-star hotels. This is not how life is, right? Evolutionary, animals and humans, we were all exposed to different stress. Food is not available all the time. Temperature and many other stressors in the environment all the time. So we introduce stress to our experiments, and this is where we see that having these genes provide some advantages. It's not on and off. They wouldn't die, but they were better off. That's why our hypothesis is that these genes help adapt faster, right? And this is the core of our sort of hypothesis and work. And yeah, Dennis and Tara can tell a little bit more, but all of the assays, they do exactly test for that.
I have a question. I'm not a scientist either, so I understood some of it. My son is 36 years old, and it's interesting when you were talking about all of this. He is currently going under a lot of testing with a neuro endocrinologist, who this gentleman seems to be really good and was very interested in learning about USP7. We have not talked to him since we've gotten all the results. I mean, I've gotten the results on my chart. But Patrick was growing up, was a failure to thrive, very, very thin, very lanky, not as tall as my other children. My other sons he's the oldest of four. It's like 6 feet and 6'2", and Patrick's probably 5'7". But when he was probably about 20 years old, he started gaining weight, and he doesn't live with us anymore, but he eats dinner with us. But during the pandemic, he was with us 24 hours, so we knew what he was eating. And he cannot regulate his weight. The other thing, he, on his own, fasts quite often. And so now I'm thinking maybe that's not a good thing for him to do. Like he works full time, and he's outside walking when he works. So he probably does anywhere between 20 and 22,000 steps a day. So he's very active. So he chooses not to eat breakfast, not to eat lunch. I'm wondering if that's maybe not a good thing for him to do.
Yeah, that's a very good question. And it's very interesting what you're saying. For example, two things. One is when you said that he had failure to thrive, he was thin and not, but then it changes at around 20, right?
Yes.
And similarly also with Hao-Young patients. You know how in our chart, and this will need to change, when we have Prader-Willi, Hao-Young, and Hao-Fountain, and this hyperphagia and obesity, it's more specific for Prader-Willi. However, you know, we didn't at the time, like we know for Hao-Fountain only a few years, right?
Yeah.
For Prader-Willi, for Hao-Young, a little bit more. Before Prader-Willi was the major, and for Prader-Willi, really this flip happens around 4 years of age.
Okay.
And for Hao-Young, these patients that have only MHL2 affected, happens later. So we thought in the beginning that this is not a problem, but now that we have also adults with MHL2, exactly similar features are coming in. So I think we need to also, as scientists, think more about it. Why is that, right? And also, it is similar problem, just it appears to come up a little bit later. So this adaptation or somehow controlling feeding and metabolic adaptation that I have enough energy is not controlled. It's maladjusted in these patients, right? So it would be great when we figure out, can we help somehow? Because they are also nervous all the time because of that, they also feel like, because the body is like, oh my God, I need to eat because otherwise I won't survive, right? This is what they hear all the time. So, but your question was whether or not the fasting, like, congratulations that he can control that. That's amazing, you know. Whether it's this good or not, let me think about it. I don't have any good advice now, but do you feel he feels okay after that? Do you feel that he is more nervous, less nervous? Because also how he feels, that should, I don't know how well you can tell or he can tell.
He's very reserved. I think he's under stress a lot, but he doesn't show it. I have thought for a long time that it was his cortisol level because he has agenus of the corpus callosum. That's what we have since he was 2 years old. We've known that he's missing the middle part of his brain. We just found out about 7 months ago about the USP7. So it was always explained to us, the world goes at a speed of 100 and Patrick's brain maybe goes at 75 or 80. So he's missing, just things going by. And so that has to put you under stress, but he doesn't show that. So I always thought his cortisol level, because he's very round face, all his weight is in his abdomen, but it came back normal. And they did a 2-day saliva cortisol and it's everything normal. Almost all his other levels, testosterone, everything else some of it's abnormally low, some of it's normal, but like right on the low level of normal. Again,
What about leptin and all of this, insulin, all of these levels are pretty normal?
Well, they're on the low, like the one that you just said that starts with the L, his value is 2.8. I think that's right below where he should be. The testosterone is below normal, but the free is right at the lowest level. So but they have dropped. They did all these tests back in 2015. And so they've compared them and they've all gone down since 2015. So I don't know, maybe as he gets older, things will start degrading.
Yeah, so this will be very interesting. So one pathway is that we found that USP7 is important and MHL2 is secretion of hormones. That's what I think Tara mentioned. That was the first pathway we discovered through this F-actin nucleation. Although this is very mechanistic, but nobody knew before, because this is the recycling of proteins in the cell. And when I started the project, it was like, okay, let's see which proteins recycle through that pathway. And we were thinking more plasma membrane, like receptors, because typically these pathways are used. And what we did, we said, okay, let's see in hypothalamus what is lost. And what we found that these secretory granule components and hormones are lost. Seems like that this recycling is so important for this pathway because hypothalamus is highly secretory. It's like an endocrine gland that secretes these hormones and needs a lot of this. Seems like that this recycling, this economical way to do that is so important. But then we also thought about it. Why do you need MHL2 in hypothalamus for that, but not in pancreas? Because in pancreas, MHL2 is not expressed. And the same pathway takes place because it's also an endocrine gland. And then we're starting to think, and this is where we are now. We try to figure out why and how we can overcome that. These are the projects that we are starting to explore, to find in different tissues, maybe even in different animals, how this evolved. Can we find something that we can overcome that and also develop method methods to screen for potential therapies that can help overcome MHL2 requirement. Because MHL2, I don't know if you, whatever Dennis was showing and Tara, it's not a major player, you know?
Mhm.
The other proteins are the major actors, but MHL2 regulates it, you know?
Okay, yeah.
And that's why whatever Dennis showed, this IQ gap 2, we were thinking, maybe this can be one of the proteins that can help. Because again, if I go back, when I was thinking pancreas and hypothalamus are neurons. And neurons are different cells. They are long. So these granules have to travel for a long path. Maybe that's also the important part that we need to address, right?
Yep.
So these are kind of things we try to develop methods, models to try to get more information about that. And we are just starting, and we just started, like we don't have any project currently on Hao-Fountain Syndrome yet, but started first collaborations to try to address that and apply for funding, get funding to do that. And I think it will be very important for like and I think Amber, you're doing already the repository of patient samples. That will be super cool then, and in critical, you know, for developing to try to address in the patients. And as you can see, every patient is different, right? It's not only one gene. It's also the background of everything else that also modulates. So these are kind of things.
Well,
So-
I think your research is wonderful. And when you were talking about the neurons, I mean, Patrick, he has agenus of the corpus callosum. So he's got the midline defect. So would his neurons possibly not be able to travel as far? Like you said, every patient is different. It's, that's pretty
Yeah.
interesting. Yeah.
And also, like Dennis can comment on that as well, right? You get hypothalamus, you know, that's like 1,000 cells is the same, and then next 1,000 are different. And, you know, when we blend everything together, we may miss what's going on. This was the best thing we could do 10 years ago. Now, with these single cell approaches, we can decompose to the different cell types. For example, one important part, Dennis, right, is microglia. This is immune system in the brain, you know, and even this seems to be changing the most. You know, these cells that are not even neurons. And why is this because MHL2 function in these cells, or the whole complex, or cells communicate, right? Among, like, if neurons are broken, they communicate it either, oh, something is going wrong, or they don't communicate what they should. So, and the other cells respond. So all of these will be important to develop targeted treatments, but also maybe symptomatic treatments, right? To alleviate some of the symptoms, to live better life, right?
Yes.
So yeah, I think that's, and yeah, whatever Tara is doing, it's also interesting. That's no, it's very hot topic now. It's known very well because in stress, translation shuts down, and only stress proteins are translated. So we try to understand, and what we see in models, translation is completely changed already in normal conditions, you know?
Yeah.
So now we try to understand which parts is this signaling stress, and also how that then interferes with adaptation, because the system is overstretched already, right?
Okay. Oh, that was great. Thank you.
Yeah, can I just because your son corpus callosum, right? It's very interesting just because corpus callosum is white matter, right? So it's these non-neuronal cells. They're like axon. There's just like axons and non-neuronal cells. So like astrocytes, oligodendrocytes, And these are exactly the cells where we see the most changes when we knock out MAGEL2 and fast these animals. So that's, yeah, very interesting.
It is interesting. Okay.
Hi,
Ella wanted to say hi to her. She's homesick today.
because I had a seizure yesterday.
So she wanted to say hello.
Hello.
Hi, Ella.
Hi, Ella. How are you?
Good.
Nice. Sometimes it's nice to be at home, right?
Yeah.
And treated specially by your mom and dad.
Yeah. All right.
Nice, lovely. Yeah, so this it's really amazing how independent your children became or are becoming. That's also so. Belinda, your son is he lives by himself?
Yes, he lives in a townhome. He drives. I mean, he gets support from us. It's only about a mile from our house. He eats with us a few nights a week. Well, probably about 5 nights a week. I mean, we support him in the sense of, bills and making sure things are paid, everything's on automatic, but he does quite well. Part of the thing with Patrick, I think that helped him. I mean, there was some negative. We moved every 2 years or every year. My husband was in the military and we moved everywhere. So he was under constant change. He went to 3 separate high schools. I don't know how many elementary. I think we when he was in school, we had moved, well, from time of birth into the time that we moved here. He was 20 when we moved here. I think we moved 15 times. So yeah.
I thought we moved a lot.
No, we moved.
Didn't get to this number.
It hurt him in his education because the continuity was not there. We started from scratch every time, but he is very self-reliant, very independent. Again, he works full time. He drives for our town and he drives a town truck. So he has figured things out pretty well for himself. I mean, I think we are very lucky.
Yeah, you should be proud of him.
So, oh, we are. We're very proud of him. He works hard.
Yeah.
He's still got challenges, but he works hard. So,
Yeah, congratulations.
Sorry, can I ask the question? Can you hear me? Sorry.
Yes.
Yes.
Hi.
Yeah, sorry, I've got a problem with my thing. Yeah, I'm not a scientist. It's kind of all new to us as well. We're trying to grasp a lot of things, but some of the things you said really interested me because so my daughter is 9 years old, and recently, for the last, I would say, 2 years, we kind of start seeing that she cannot control the hunger, and it's going worse and worse, and to the point that we all have to carry on looking at her what she's eating and she keeps telling us she is hungry. So now we actually ask if there is anything, and she was a part of a study in Germany, and then we actually got the result, and one of the results shown, I mean, this was all in Germany, so I do not know that much.
Dr. Schaaf?
Yes.
Mhm.
So she was part, but obviously this is all in German, so I was trying to translate it somehow, but somebody said that one of the indicator, which said something about insulin resistance, something like that, she had really, really high. So now I hear that in this report, that something about not regulating insulin. And so this is what I read as well about that, sometimes people can be very hungry when they cannot regulate insulin. Is that what you kind of...
Yeah, exactly. No, I think it's very interesting because there are many mechanisms. We are now really diving into this appetite regulation, because in Prader-Willi syndrome, that's the main problem. Like they just recently did study with the families, and family identified this is the major stress in their families, because they need to lock everything. They need to be on the look all the time.
Yeah.
I'm listening to both podcasts. I don't know if you're listening about Tess. She has Hao-Fountain Syndrome,
Yeah.
right? And he mentioned the same thing. Like they also started to see that in Tess. And as I can see, several of you guys are mentioning that also in Hao-Fountain Syndrome. So the regulation of satiety is really complex, right? There is leptin, insulin. So it's really the satiety regulation together with the metabolic homeostasis of metabolism connection, right? Including then insulin resistance that can contribute then back to also appetite regulation. So we will definitely focus on that, and other people are doing that. And also, this is exactly like first time when we got this data, I asked students a couple of times, are these groups correct? Is this correct? Did we mess up something? Let's double check. Because we couldn't believe really that what we see, right? How these animals that have food available and everything is normal, they are just knockouts, but they are almost the same as the fasted ones, you know? This is interesting, right? And the cortisol levels are also, oh, cortisol levels. Okay, let me think about it. Now back to Belinda, your comment. Not necessary cortisol level is up in those knockout mice, right? Dennis and Tara, remember the difference? Cortisol level is not different, you know what I mean? However, the hypothalamus is different, independent of corticosterone. That's interesting, you know what I mean? That's super, because this is something, Tara, if you remember, that's part of your proposal also. We were thinking maybe cortisol levels are different, even in, but of course, these hormones are also very difficult to study. I will tell you why. Because they are so short-lived, it's so difficult to get the experiments and fixation that, and everything changes them to get good difference. Maybe time points matters, right? So, but still, I think this is because we always measure fasting, of course, corticosterone is through the roof, but there is no difference between wild type and knockout. Although on molecular level, the tissue is different, you know? That's really good points. I think these discussions are really interesting because give us ideas, you know, what we should focus on, what we should really now,
Because from the hormone levels, this is also, now if I can think about this, it's like, I know it's very early, but I actually, this is, like I said, all new to us. But yeah, she's a 9.5. But for last 1.5 years, she's beginning to actually have hormonal changes. You know, we can see like some hairs, and yeah, basically it looks like it's actually already starting slowly. And that's previous to that, it was fine. She had 3 meals a day, you know, and some snacks in between. But now it's like she could eat and then literally someone will be eating and she's like, I'm hungry. And it's like, goodness me, now we need to start almost, and you know, and then when we say no, it's like end of the world. So yeah, that's kind of, but when we measured her sugar level in just a normal doctor clinic, they said, oh, it's fine, you know, like her sugar is all fine. So now I'm thinking maybe we need to do some further tests with that insulin and how she reacts to that.
yeah,
But
yeah.
and all the and yeah, she's gaining weight as well, and all her, like, let's say, fat, it's all about abdomen. Like her legs and her arms are all great, but it's just all about the belly. It's like huge belly she's got, and it's like it's hard.
And actually, you know, if we go back to MHL2 mice again, and I think important thing, we need to really determine MHL2 levels in Hao-Fountain models and in patients, you know, that's an important part. Also will be very helpful to also understand or even like combine, right? If we find something that we can help, and definitely help both. You say, this MHL2 lean versus fat mass. Exactly, this is what you see in MHL2 and Schaff-Young models, you know, and patients, that fat is accumulating. And now I'm thinking another thing we need to think, like overall in other tissues, what is going on, right? What are the pathways there that we could, you know, that are potential for intervention, even though it's not the primary cause, might be secondary, you know? So definitely very, very informative information from your side. And
Thank you.
we'll
You're doing great work, by the way. So thank you very much for sharing this with us.
yeah, we are trying to do our best, and we need to publish that to get it out. And also we started to collaborate also with another scientist in Texas who is also developing in vitro models, and actually it was amazing. I think she saw a similar idea with knockouts and fasting in a cellular level, and definitely that's our plan now to go really to find the molecular mechanisms behind.
Just one more question. So you mentioned something about immune response as well. I mean, because like my daughter actually is now also, looks like, I mean, to be honest, through the years she had, it's just now changing from one part of the body to the other. And it's like literally, basically, she's all the time ill. And it's like in the last 9 months, she had a pneumonia out of the blue, where before she had always tonsillitis and, you know, ear infection. So it was tonsillitis, ear infection, and now it's pneumonia. It's just growing, and it's like there's nothing really they can find which causing it. It's just out of the blue, and it's like she's just really, really weak. It's like nobody else will be ill except her. So, or she's undergoing some tests now for immune. But I heard you mentioning something about the immune as well. So that's interesting.
So yeah, that's very good point also to and we need to go into the literature back again, but just to see overall immune status of the like models, as well as whatever it's known, and in patients. But what we see, for example, even in hypothalamus, that seems like that the immune system is activated, you know, because these microglia, these are immune cells in the hypothalamus. And not only our data, also data from other scientists are suggesting that this, you know, it is immune, somehow immune system is activated in those tissues. And these can contribute also to the phenotype, you know, and to aggravating the situation. That's a very good point. However, like very little is still known the molecular mechanisms and how this works together. But with these single cell approaches, exactly this is what we want to do, and like we can do as a community, right? To understand different cell types, what do they do, how do they contribute, including the immune cells. Because in general, like in the brain is more or less immune privilege organ. So because of the barrier, blood-brain barrier, immune cells mostly don't go through so easily. However, hypothalamus is the most open, you know, and that's also one important part of it, right? And So definitely an important avenue to also investigate, and maybe even potential for alleviation, you know, some anti-immune approaches. Because eventually what we also want to do, right, and Dennis started, we didn't talk about it, but also to try to investigate the gut microbiome, because the whole body is connected, right? And there is a lot of, in particularly gut-brain connection is now very well known. And I don't know if you know, Rett syndrome is one of the neurodevelopmental syndromes as well. And one of the probiotics was shown that somehow is helpful in those patients. So this brain-gut connections, it's also another avenue that we would like and others investigate, and maybe, you know, the intervention is maybe possible, right? Because you can maybe do something with probiotics, with food, etc. So these are, but very early stages. Don't, you know, don't quote me on anything yet, but definitely very interesting.
Yeah. And
Thank you.
so Ella has IgA deficiency. So for Anna, that might be something you would look into if they do that, the IgA deficiency. I mean, we knew that at a very young age because she had some food allergies and they did a lot of testing, and then they've retested it a few times to see if it's still, you know, in effect. And, you know, it's been like 0, her IgA.
So
And she
she's
used to get sick all the time, every cold, every single thing that would go through. She had IgA, and there was another USP7 child that had an immune, what is it when it's like multiple immune deficiencies? Like she had like the IgM, the IgG, like all of those. And unfortunately, that child has passed away, but she had multiple immune deficiencies. I don't remember all of them, but she had many.
Yeah,
Well-
and you know, USP7, of course, it's expressed in every cell, right? And USP7 has so many functions. This could be major 2 related, could be unrelated, could be both, right?
Yeah.
That's
Yep.
definitely
And you know, Ella, when she was a baby, she did have a lot of gut issues, and she had, you know, like when you get like those overactive bacteria, like she always had that going on and needed probiotics for that.
Yeah, Isabel. I mean, Isabel is kind of she was since she was baby, she was always ill, like much, you know, we were numerous times. Yeah, I was giving myself. I was giving her obviously probiotics and stuff. She does have multiple allergies as well, like quite bad. But she also have a constipation, so we have to give her. But now recently as well, what we found out that she's got this. I don't remember what um, it's a enzyme resistant to antibiotics in her urinary tract infection. So kind of this kind of things, and it's just kind of like it's adding all the time. There's always something new, but something with immune and with all different things. So it's kind of hard because obviously not a lot of doctors knows about this. They don't even want to look into that maybe there is something connected all together as like a bigger picture. Everybody wants, like here where we live, wants to treat the condition then and now rather than look into. But thank you. I will definitely look into this. What did you say? IG,
IgA. Yeah,
IGA.
IgA deficiency.
Yes.
And that is like definitely like colds, you know, pneumonia, bronchitis, like all of those things. And also some gut issues can be from caused, you know, from that too.
Cool. I will definitely check that. Thank you.
Yeah.
Anna, I was going to tell you everything you said about your daughter. It's the same with my son, the gaining weight in his stomach and in his head and stuff. He was sick as a baby, ear infections, had his tonsils and adenoids removed because he kept getting sick. has had multiple pneumonia, bronchitis, and no one else is sick. Nothing has changed, but he will get that. He recently, because he got pneumonia so much, they did a swallowing test to make sure, and it was normal. So they don't know why he keeps getting pneumonia. His glucose and his sugar, his insulin is always perfect. The one thing that did come up, he has fatty liver disease, and it's gotten significantly worse. And they did a GGT test. I'm not really sure what that is, but it helps them determine what's causing it. But everything, so I just want you to know you're not alone. You're describing my son exactly.
But you know what? It's obviously my daughter is 9. So it's like, it's really, really nice to hear that your son is, is managing. I know it's not on his own because you support him, but it's, it's amazing to hear that, you know.
Yeah. And when he was 9 years old, we didn't think he would ever live on his own.
Yeah.
So we had a doctor, a neurologist, in North Carolina. We always came back here everywhere we lived. And he told me when Patrick was, I think, 6 or 7, he said, don't ever let anybody tell you he can't drive. Don't ever let anybody put any kind of restrictions on him. He can do whatever he wants. And again, when he was little, we never thought, I mean, it's just, and really, to be honest, I think the most growth has come since he's been 20.
Is it? Oh,
Yeah, he has done really well. So yeah, he struggled an awful lot growing up, but so.
Oh, that's really, it's really good to hear. I'm really happy for your son.
Yeah, well, I mean, it's scary when your children are young. It's scary. So.
Yeah, I mean, she's doing amazing. To be honest, when we went to Germany, um, Dr. Schaff, he said that she is actually, you know, in that respect, because she talks, you know, she's struggling with she doesn't talk like all the other kids in her age, but she does talk. She's got she does obstacles in life, but she's very stubborn. And it was really funny because one of the questions on this phenotype study was, does your child have manipulate behaviors? I don't know if you ever seen this kind of questions. And we laugh at this, but then actually from then we thought she does actually have manipulate behaviors. And this is actually, maybe that's what helps her as well. So I'm really, really, it's really nice to hear, you know, that he's doing so well, actually. And yeah, an amazing information from all of you guys. Thank you.
Yeah, thank you for listening to us and telling us your side of the story. Very important for us.
Yeah, thank you so much. I just thought this was great, and the conversation after was great. Everything's just wonderful. Thank you so much.
Yeah, thank you for having us, and we'll be in touch. We will try to get funding and work together to get funding for our research and move forward with that. So definitely very interested to help bring our piece to the mosaic. Right.
Great. Thank you
Thank
all.
you.
Thank you.
Great.
Thank you.
Thank you.
Good seeing you.